Good evening, everyone. It's Doug McConnell, and I'm the co-founder of A-Long Swim. We are so grateful for everybody to having signed in this evening. It looks like there are people joining even as we speak, but I want to be mindful of everyone's time and so forth. The A-Long Swim is the host of tonight's webinar. Obviously the sort of queen pin of the A-Long Swim webinar series is Hande Ozdinler, who is the head of the Ozdinler Lab. That is all of the proceeds that A-Long Swim raises in turn supports. One of the reasons that we were so gung-ho about supporting Hande, and now for 10, 11 years, is because in our very first meeting with Hande, she really was supporting and emphasizing the importance of collaborative research. And one of the things about marathon swimming is you learn pretty quickly. You may be the only swimmer in the water, but you are nowhere without a team. And Hande had really kind of taken that same approach with so many of the projects that she pursued and so forth, and now 10 years on have really become flourishing projects. So one of the things that we discovered was that in addition to the collaborations that her lab has had with ALS research centers around the world, here there were collaborations right within the four walls of Northwestern University itself. And starting a couple of years ago, some of those collaborations were formalized with the CLP group, Chemistry for Life Processes, which happens to be at the Evanston campus of Northwestern. Not only stitching together those two campuses, but really some of the very, very profound expertise of these members of CLP from Evanston. The two researchers that we've invited and are really honored to have this evening are part of that, and are going to talk a little bit about the very special work that they do as it relates to translational research and I'm going to say this wrong, high throughput research that we're going to learn all about this evening. So my job is to get out of the way and to introduce Curt Horvath, who's kind of a key player in that whole process. And Curt is the one with beautiful Lake Michigan behind him. Before we do the handoff, Curt, I want to invite people really to two things, and I'm reminded of Lake Michigan right behind him. 2 years ago, we swam from right where Curt is sitting in the picture to the downtown campus of Northwestern, and we're inviting people to do that same swim, Northwestern at Evanston to Northwestern in Chicago. That's going to be in mid-September. A warm-up swim that we're having for that is day after tomorrow. That'll be up in Winnetka, right in Lake Michigan. So for additional information about these wonderful swims, please go to alongswim.org. Enough of the commercial. Curt, you're on.
Thanks, Doug, and it's a pleasure to be here and with Sara and Hande to talk about high throughput screening and core facilities in research. First of all, my name is Curt Horvath. I'm a professor in the Molecular Biosciences Department here on the Evanston campus. And one of the jobs that I do, in addition to research and teaching in my own laboratory, is that I'm the facility director for the Northwestern High Throughput Analysis Laboratory, or HTA or HTAL. And one of the things that we do in science, we make discoveries in our own lab doing basic research, but sometimes the thing that really drives your project forward to develop into more of a biomedical application is to access technologies and expertise. And as you mentioned, Hande has been really good at doing this collaboration with other scientists. But a lot of universities have invested significantly in bringing the cutting edge technologies and well-trained expertise into things called core facilities that would be able to provide unique resources that a number of researchers can use, but they don't have to buy that stuff and have it in their own lab and hire somebody to run it. And so the high throughput analysis lab is one of the core facilities we have. It is part of the portfolio of core facilities run by the CLP Institute, Chemistry of Life Processes Institute. And they also have facilities for proteomic analysis, microscopy, many other applications. So one of the questions we're going to address today, Sara, in fact, will talk about this, is what exactly is high throughput and how we use it specifically to take care of research projects. And one of the important aspects is really in the application of discovering drugs. And as you know, there are diseases like ALS and many other diseases that people suffer from that there are no drugs or treatments available. It's not for lack of research, but for the complexity of those particular diseases and unraveling the biological realities that underlie the causes and consequences of these various complicated diseases. And in particular, neurological diseases are particularly difficult to study. So we try to make some laboratory approaches to develop model systems that have the appearance of the disease. And then we try to throw as many different conditions and chemicals and inhibitors at them as we can in order to find ones that work and ones that don't work, to give us a clue. I guess it's a little bit like if, since Doug is out there swimming for a long swim and raising funds for research, if we could clone Doug, instead of having one race by him, have 96 races or 384 races at once and reap the benefits from all of those opportunities at once. And that's really what the high throughput analysis does for us in research, to help us take the discoveries we make every day in our lab and turn them into something that may be more applicable to the clinical setting. And we call this process translational research, or translation of basic discoveries into biomedical applications. So that is really, in a nutshell, what the core facilities are about. And at this time, I just want to introduce again Sara Fernandez Dunne, who is the managing director of the high throughput analysis core facility. Sara is really the person with the expertise and the knowledge of the instrumentation who gets together with basic scientists like Hande or people in her team, or researchers in various different fields of study throughout the university, both here in Evanston and down at the medical school. And in fact, our facility is available to all the institutions in the Chicago Biomedical Consortium area. So that's a shared facility for all of the institutions locally to use. And I think Sara is going to take us through a few slides to describe exactly what it is that we have in terms of instrumentation at the core facility and how we use it to tackle some different research applications.
Okay, thanks, Curt. And thank you, Kate and Doug, for organizing this webinar today. Let me just get my slides up.
You can share your screen, right?
Yeah, I'm working on it. Here we go. Can y'all see it? All right, so I'm going to be telling you a little about the HTA and what we do. And in the process, I'm also going to go over what high throughput screening means. Curt touched on it a little bit, but I'll go into a little bit more depth. And just as a side note, some of the slides that I'll be showing today are actually from a lecture for a graduate level science class here at Northwestern. So I'm sure y'all didn't think you were going back to school today, but hopefully I don't put anyone to sleep. And in the end, I do hope you all learn a little bit. And so with that, oh, and this video you're seeing is a little preview. So you'll have a visual reference of all the things I'm talking about.
And here it says 200,000 molecules available. So that's also very important.
Yeah. So Curt covered the first couple bullet points here. So I'm just going to skip down to our mission, which is to provide research with tools and expertise for high impact, large scale biology. So in simpler terms, that means we do something called high throughput screening. And specifically, it's part of an early drug discovery campaign. So what does high throughput screening mean? A simple definition is it's the use of automation to test thousands to millions of conditions in parallel for biological activity. And it can be done at the level of a model organism, like we use things like nematode worms, yeast, zebrafish, down to the molecular level. And so in order to accomplish these massively parallel experiments, we use something called multi-well microplates. So instead of working in individual tubes, we do the experiments in these multi-well plates. And these plate formats were standardized back in the 90s by a group called the Society for Biomolecular Screening. So now we can be confident that all these SBS standard microplates are compatible with all the different types and brands of lab automation that we use. And so 96 well plates were the first to be introduced, the one here on the left. And since then, 384 well and 1536 well plates have been developed. And so for me, even though I use them every day, I still find it mind-blowing that with this one plate, I can do 1,536 experiments in parallel.
Sara, what's the volumes on those plates? Each one of those wells, how many microliters are in it?
So in microliters, the 96 well plate is usually 100 to 200 microliter assay volumes. The 384 is like 20 to 50. Although there are specialized plates, all of these have specialized versions. So there's like low volume plates that have even lower. And then the 384 is usually 1 to 5 microliters. So that's 1,000 of a liter.
So it's less than a drop. And just the impact of high throughput screening, when you think about it, it's not only that you can do a lot, but you miniaturize everything. Yeah. And can use these incredibly small volumes to get meaningful data on chemical inhibitors and things like that.
And I think that's why you need automation, because precision is very important, right? So you wouldn't be able to do this by hand and filling like 3,000 wells all at the same time.
Absolutely. I've gotten to the point where I can work on the 384 well plate manually, which, only because I've used them so much, but the 1536, forget about it. You can't keep track of where you're pipetting on those plates.
And in the typical lab works with the tubes on the left, the individual microcentrifuge tubes. And you can't even compare those individual reactions to what you get on these multiple well plates. They're so miniaturized and specialized.
Yeah. So I guess the next question would be, why do we use high throughput methods? We just sort of talked about it. So you can imagine it will drastically decrease the time it takes to complete a project when you're probing a large number of conditions. And then it also decreases the cost because the volumes of the experiments are miniaturized, which saves us like precious samples or expensive reagents. And because of the time and cost savings, researchers can increase the scope of their work. So, and then plus the use of automation can improve data quality and reproducibility. This is all stuff we sort of just covered. So humans are quite capable, but the robots, they won't get distracted by a lab mate asking where something is, or they won't get tired because they stayed out too late last night. They'll just, do what they're programmed to do every single time. So you might be wondering what types of experiments do we apply these high throughput techniques. So we already covered drug discovery, but also this can be applied to different basic science research. So in the different omics fields, proteomics, genomics, metabolomics, there's tons of them nowadays. And then there are a bunch of other fields. Really, HTS has it's become pretty much a universal technique in science laboratories. So here in the HTA, we perform two main classes of high throughput experiments. So they're based on the experimental modifier. It can either be genetic or chemical. So genetic screens use something called siRNA or shRNA or viral vectors or CRISPR, everybody's probably heard of. And then on the other hand, chemical screens use small drug-like molecules. And so generally speaking, genetic screens are trying to elucidate the pathology of a disease, while the chemical screens are early steps in drug discovery. And they're usually targeting a specific protein or a phenotype that's known to be part of a disease etiology. All right, so I'm going to tell you a little bit about the instruments we use, and specifically the ones we have in the HTA. They fall into three categories. There's liquid handlers, plate handlers, and then detection instruments. So liquid handlers do just that. They move liquids around. They range in throughput from manually operated single channel pipetters to fully automated liquid handling robots. And so when we're talking about high throughput, what we do in the lab, we're using these instruments on the right side of that scale. And so here's some pictures of some instruments that we have in the lab. So we have a couple big automated, what's called pipetters, big automated pipetters. And then they also have like a robotic arm to move plates around. And they can do really complicated liquid handling tasks like walk away. We can just have it run the assay for us. There's something called bulk dispensers here on the bottom. They can quickly pump liquids to fill microplates with a single reagent going into all the wells. Our pride and joy is our Echo acoustic liquid handler. So the Echo actually uses sound energy to eject a 2.5 nanoliter droplet of liquid from the source plate into a destination plate. And it provides us with like a rapid touchless liquid transfer. And it's really a vital instrument for us when we're working in 384 or 1536 well plates, when the total volume in each well is already only a few microliters. So any addition of reagent needs to be a really tiny volume. And so with our Echo, we can add any increment of 2.5 nanoliters.
Yes, so Sarah, nanoliter, I mean, it's very hard to understand. So that it's, micro, right? And then you go, right? So imagine that.
Yeah.
But then how can you plate cells or neurons or, because there will be some cells to do the assays with, right? So you move the chemicals, but how do you plate the cells?
So usually with cells, we'll use those bulk dispensers because those are usually in microliter volumes. And also, I mean, it depends on the cells. The cells you use are more precious. So we have some cells that we can grow liters of it, it's much more easy. The bulk dispensers they have a really high dead volume. So the dead volume is what the extra amount of liquid you need besides what you're going to put into the plate. And so, on the next slide, we have another instrument called the Mantis. So that has a really low dead volume. So that's something we might use and we do have it inside a hood, so a biological safety hood. So we can use it for cells, and it has a really low dead volume. So it'd be something we might use for your cells since they're so precious.
And one thing it occurs to me that maybe people in the webinar may not be so familiar with, that just to say that in life sciences research, biomedical or molecular biology, biochemistry type of research, what people do in the lab all day is transfer small amounts of liquid from one tube to another, and then mix it, heat it, put it into a reader, do something with it. And so hours and hours are spent transferring large amounts of liquid into small tubes, and then small amounts of liquid into another tube. And as Hande was saying, what are these volumes? Well, a milliliter, that's a cubic centimeter of water, right? And we know what a liter is from a liter bottle. So, it's a 1,000th of a liter is a milliliter. But these nanoliters, that's a millionth of a liter. It's less than, the spittle coming out of my mouth right now. It's just a tiny drop. You cannot really see it. And if you try to transfer that manually, you will not be accurate. There's just no way. So this sonic transferring is very precise. It's a super high-tech method, and works by physical principles that have been completely miniaturized just for this application. And it enables us to do so many experiments with a finite number of compounds that we have in the lab to do drug screening and things like that. So it just boggles my mind that we can do hundreds and thousands of these reactions at such a small volume.
Definitely.
Yeah. So this is the mantis. Looks like a little bit like a praying mantis when it's dispensing things. And then we also have the mosquito. So the mosquito uses these tips that actually have metal pistons to dispense. It's also in the nanoliter range, anywhere from 0.1 to 1.2 microliters. And you may have noticed there's a bunch of insect names. This is not the only insect liquid handlers out there. I'm not sure why there's so many insect names and for liquid handlers, but anyways, it's probably just a trend that sort of stuck. All right.
Sounds like Marvel superhero.
Yeah, there you go. The mantis and the mosquito. All right, so I'm not going to show you our plate handlers, but we do have plate handlers in the lab. So i.e. that's just, moving plates around from instrument to instrument. And so just like the liquid handlers, they are on a scale from a manual to more automated. So, manual being just picking up the plate with your hand and moving it into an instrument. And then, then there's something, kind of a center or a middle ground where you might have an instrument that just feeds a single instrument. And then there's these fully automated systems where, after the system has been set up, there's no human input at all. So we are actually here in the HTA, our plate handling methods are much more on the left to center end of the scale. These big fully automated systems are really only seen in big pharma. They're kind of uncommon in academia. And then for the detection instruments, these are what are actually measuring, the biological phenomenon that we're interested in. So the first kind of common instrument is called a plate reader. They are used to detect biological or chemical events in microtiter plates. That's their purpose. The different detection modes that we use is there's absorbance. So that's how much light at a specific wavelength, the solution will absorb. And then there's fluorescence, intensity, and luminescence. So you're all probably familiar with that. And then there's some, specialized variations of those. And then we do have one unique plate reader. It's made for fast biological reactions. So it actually has an onboard liquid handler inside the box, and it can read the entire plate at once. So it can give you that 384, wells all at the same time. So it's super high throughput, and it's made for very fast biological reactions that, if you add a reagent, it's going to happen within seconds. So you don't have time to sort of wait for the plate to be put inside the machine. It has to be read immediately. And then we also have what's called a qPCR machine, which we actually I don't know if anybody knows what PCR is, but we don't use it for PCR. We use it for another method, another assay for, it's called, it's protein unfolding. So we're looking at protein. All right, and then we also have something called a high content imager. So these are essentially microscopes inside an automated platform, and it enables automated image acquisition. So, and then included with any high content imager, you also have some really powerful automated image analysis software. So high content is a subclass of high throughput screening. It combines that automated imaging with quantitative image analysis. And it does enable some readouts such as cell morphology and protein localization within the cell. So that's some information you can't get from a plate reader, hence the high content in the name. Here's one example of an experiment that's enabled by our high content imager. It's called a 3D spheroid screen. So when cancer cells are grown in a gel in the microplates, they'll essentially grow into tiny tumors or spheroids, we call them. And so we use that assay to test for compounds that can kill cancer cells. So with this experiment on the left, there's a blue stain that was added that allows us to mark the entire spheroid. And then a green dye is added to mark the live cells. And then there's a red dye that marks dead cells. So we can get a % live and dead measurement in addition to just the overall size of the treated and the untreated spheroids. And then over here, we have on the right side, we have an example of a spheroid experiment using no stains, but we just use the transmitted light of the microscope to visualize and measure the size and the number of the spheroids. And then this is a good example of morphology measurements. So it's a neurite outgrowth experiment. So this is a really common experiment we do here in HTA. And so on this little top left panel, you see the fluorescently labeled neuron in green. And then the red here is the mask that's generated by the analysis software. So not only does the software count the number of neurons, it can give you the length of the neurites, neurite thickness, intensity, branch points, and there's a bunch of other parameters that it can give you. And so this is the assay that we do for Hande. I'm going to stop here and hand it off to let her talk about her work with ALS. But before I let you go, I do want to say that I love to show off the core and our instruments. So I'd like to encourage anyone who wants to see them in person to go ahead and contact me. My contact information is there. And I'll also put it up in the chat so people can contact me if they want to come see the core.
Hey, Sarah, before you drop off and hand over to Hande, there are actually a couple of questions in the chat for you. Can I read them?
Yes, please.
So Nicholas Gerst is asking, how do you know that all the compounds results are equivalent? Some compounds are not very soluble. You don't want a negative result just because the compound was not in solution during the assay.
That's a great question.
I knew you'd like that.
The answer is that our compounds are actually sort of intelligently designed. So to make them more soluble, they have, we can't guarantee that, we do only do it at a specific concentration, the maximum concentration, so that we avoid possibilities of them being, crashing out of the solution. But also we spent a lot of time, the medicinal chemists basically went through available libraries of compounds and selected compounds that are, have these qualities that make them better candidates. And one of those qualities is solubility. And then
so maybe more compatible with a biological system.
Exactly.
As opposed to dissolving in a solvent for maybe use in an in vitro assay or something.
Right. But we do have to be cognizant of that potential. And we do have a maximum concentration that we'll screen at that hopefully, most compounds will be soluble at that concentration.
Great. And Nicholas has a second question. a lab looking for your high throughput expertise may have an assay. Can you usually use it, or do you have to adapt it to your technology and equipment, like reducing the volumes?
We always have to go through an optimization before we start any kind of screening. And sometimes it's more time consuming and more work than the actual screen itself. Once the instruments are sort of set up to do screens, they're kind of doing all the work. But the optimization steps, yeah, we have to make sure that the assay is good, that it will work. Number one, that we have our instruments set up correctly, and that we have a quality assay that we're running. So there's a number of mathematical tests that we have to run them through before we'll actually start screening.
I know from our own screening project that we've been working on with you over the last couple of years, we probably spent 2 years developing the assay.
Yeah.
And then we did the assay in a day.
Yeah, that's pretty fast.
Yeah, like think 9 minutes and then execute once, 9 minutes, execute 1 minute type of thing, right?
Exactly.
And I think we are on that time scale now. We are trying to optimize our assay, but once it's optimized, I think we're going to have very robust and very reproducible results. But optimization of the assay really takes the time, right?
Right.
So let me see if I can share my screen. And I think, listening to you, people understand why we work together, right? And we're working on ALS, a rare disease, and brain is complex, but the neurons that degenerate in the brain make up maybe 0.0001% of the whole brain, right? So the number of cells that we can use in our assays are very limited. So we can't use, large big wells. Our starting material is very, it's very precise and very small volume. So we have to go small. And but it's not also feasible to do high throughput. In our case, I think we will have to find a way like mid-high throughput type of thing. But we are very lucky to have CLP and you as our colleagues to move this field forward. And I'm also thankful to Along Swim for supporting the initial phases of this research. And now our collaboration actually was funded through DoD, and we have 2 years to optimize this and hopefully to get good results, right? And I think Chris, Baruch, and Omar are in the audience now, and they are the ones who are actually working in the lab, trying to generate the tissue cultures from diseased neurons, healthy neurons, and add different compounds, try different compounds to see which improves their health and which improves better axon outgrowth, branching, arborization. Those are the readouts for the upper motor neurons. And I think we are very lucky to have colleagues like you and to have centers like the High Throughput Center at CLP. And I think CLP is the innovative engine for Northwestern. And so I'm very happy to be with you and on this webinar. And as the topic says, the high throughput for rare diseases, is it possible, like drug discovery, is it possible for the rare diseases? And as you know, usually the drug discovery field or, the drug companies are interested in diseases that affect a large population of patients like Alzheimer's, Parkinson's. But when it comes to ALS, HSP, PLS, because the number of patients are not very high, they don't show too much interest. And plus, the translation has been a problem in the past, especially in the field of ALS. Curt, correct me if I'm wrong, but there were so many ALS clinical trials and they all failed. Like there were 36 of them failed, one after the other. So then, even the drug companies say, tell me more, give me more biological insight, give me more translational data. And previously, people thought if we can improve the lifespan of an ALS mouse model, that's going to improve the lifespan of an ALS patient. And guess what? It didn't work out. So then we realized translation is not at a mouse level, Then people said, oh, maybe mouse is not a good model. Maybe we shouldn't trust mouse. Maybe, you know, then they started saying all the bad things about the mouse model. So we realized actually that the translation is at a cellular level. And what I mean by that is the cells, the neurons that we have in our brain, the upper motor neurons, let's say, at a cellular level, can be very similar to the upper motor neurons in very well characterized mouse models. So we don't really have to compare mice and human anymore. We just have to compare cells with cells, okay? And here the upper motor neurons are located in the brain, and the movement starts in the brain. So their degeneration is seen in not only in ALS patients, but also in HSP patients and PLS patients, which are even more rare than ALS. And if you think about the spinal cord injury patients, the long-term paralysis observed in spinal cord injury patients is also due to their upper motor neuron degeneration and upper motor neuron loss. So these neuron populations are critically important for at least 3 rare diseases, ALS, HSP, PLS, and also the injury, spinal cord injury. And yet, even for those diseases, right, if you look at the drug discovery platforms, none of them have incorporated upper motor neuron health as a readout. Can you imagine? So they're using unrelated cells or maybe some neuronal cells, but none of them have ever used upper motor neuron survival as a readout. So their goal is to improve the upper motor neuron in the patient, but they never ask the upper motor neuron whether the compound they move forward in clinical trial is appreciated by the upper motor neurons. So we decided that this should change, and our ideas were appreciated by the field, and we were invited to write drug discovery reviews, 2 reviews actually. One of them is that it suggested moving forward in clinical trials for ALS, we really have to ask the motor neurons, not the mouse survival, but the motor neurons itself. And the other one is the incorporation of upper motor neuron health in ALS drug discovery was pivotal, critical, and required. But how do we do that? It's not easy, right? It's not easy. And the other thing is, where is the translation? Because anybody can have experiments, anybody can have results, but then how will our findings translate to human condition? So we know mouse and human are different species, okay? So mouse survival is not equal to human survival, okay? But then what? But if you look at the upper motor neurons in patients and the upper motor neurons in well-characterized mouse models, they're pretty much similar, means they're large pyramidal neurons with big apical dendrite, long axon, they're located in layer 5, they're excited neurons. So they have very many similar characteristics. But if you go even deeper inside the cell, you realize, oh my God, their mitochondrial degeneration is exactly the same. The inner mitochondrial membrane is broken in the human, in the well-characterized mouse models. Endoplasmic reticulum has all these problems in the human and also in the upper motor neurons of the mouse models. Nuclear membrane has problems in the human and also in, so then if I show you 2 slides, right? And these are EM images prepared by Mukesh Gautam, and he's a Longswim fellow. And if he shows you 2 slides prepared from upper motor neurons in one species and upper motor neurons in another species, you wouldn't be able to tell the difference. So then we realized the translation is actually at the cellular level. Like our goal must be to improve the health of these diseased upper motor neurons. But it's not easy, right? How do we isolate them? How do we see them? How can we make them? So this was actually published many years ago, but we made the first reporter line to upper motor neurons. In this model system, the upper motor neurons that degenerate in all these patients like ALS, HSP, PLS patients are fluorescently labeled. This is very important because now you can distinguish upper motor neurons from other cells and other neurons in the brain. So then we can actually see them. So once you see them, you begin to distinguish them from the other neurons and cells of the brain, right? But then it comes even better. because then you can actually cross these reporter line with many of the well-characterized mouse models to generate upper motor neuron reporter lines of different disease models. So that, for example, you have an upper motor neuron that becomes diseased due to misfolded SOD1 toxicity, an upper motor neuron that becomes diseased due to TDP pathology, an upper motor neuron that becomes diseased due to, let's say, profilin mutation, alsin mutation, spastin mutation, and so forth. And these are all pathologies and mutations observed in ALS, HSP, and PLS patients. This is a representation of a rare disease. So now, for the first time, you have fluorescently labeled disease neuron that degenerates due to different mechanisms. So you not only bring cellular clarity, but you also begin to identify different mechanisms, right? And I need to emphasize here that even though these diseases are rare, there's an immense heterogeneity among the patients. So it just gets even more complicated because there are very few patients, but patients may become diseased due to different underlying causes. So then drug discovery was already challenging. Now it becomes almost impossible because one drug is not going to cure them all. And plus you also have a handful of patients, right? So no drug company is going to be interested in them. But how can you pull drug companies into this quest and make them more interested? I think one possible way is to develop a mechanism-focused drug discovery so that you begin to classify patients based on the mechanism. Say, for example, this patient population develops a disease due to TDP pathology. This, let's say, due to oxidative stress or lipid homeostasis or cytoskeletal problems, right? So you have different patients. Then the drugs are developed based on a given mechanism. So then you can say, all right, let's say better inclusion criteria, this patient would benefit better with this drug, this patient would benefit with this drug, and that we actually improve our success rate. So this is hypothetical, but it's also, it's becoming a reality. Curt, you have a question?
Well, I just wanted to remember to comment that as we were looking at these slides before, that I was just seeing your mechanistic hypothesis and mechanistic model for drug discovery just seems like it perfectly lends itself to the high throughput drug discovery method. Each mechanism itself has to be broken down and challenged with the library of compounds, and then it will be different compounds that score for mechanism 1 versus mechanism 2, 3, 4, etc. And then ultimately, maybe a patient gets characterized, can take compound 1 instead of compound 4, or perhaps there's a combination therapy that will be indicated so that you can cure more patients by giving a drug cocktail.
I can't agree more, and I'm going to show you in some data to confirm that this indeed a mechanism. So because in our GFP model the upper motor neurons are GFP, when you put them in culture, in dissociated motor neuron cultures, you can see which one is the upper motor neuron. So this is very cool because in the mixture of all these cortical cells, you can point the upper motor neuron. So then when you add the drug, you basically watch them grow. If they are happy, they will send long axons, they will branch arborize, but if they're not happy, they're going to shrink and they're going to die, right? So we have a readout. The upper motor neuron health becomes like a readout. And we actually, in a plate, you can see the GFP neurons, and after the immuno, you can see the astrocytes, microglia, so you can see which one is which, but you can definitely define the upper motor neurons. The other thing is, you can look at let's say, here's an upper motor neuron, it says it like it's running away, but here's an astrocyte, and here's another astrocyte. So you can see the astrocyte and motor neuron interaction also, and this could be another readout for neuron-astrocyte interaction. And I think our ability to see these upper motor neurons, both in vitro and in vivo, allowed us to identify the MU9 together with Dr. Silberman as the first compound that improves the health of upper motor neurons diseased by SOD1 toxicity and TDP pathology. Now, coming back to your points, Curt, yes. So when we add NU9 together with, let's say, Riluzole or Adaravone, 2 FDA-approved ALS drugs, in the SOD1 model, we showed that they actually supplement each other so that there's an increased improvement, right? And it is true that maybe in the future, those patients will be getting cocktails of drugs, not just one drug. And I don't think one drug is going to help everyone. We will have to develop personalized medicine approaches so that each patient may get a cocktail, and those cocktails may be determined in the high throughput assays, that you develop, so that we will know how these diseased upper motor neurons respond to these cocktail treatments or individual treatments and so forth. So coming back to this, I don't think I should.
Sorry to interrupt you, Anna. I'm just so excited to hear about your ideas on this. And I was just going to say that your collaborator, Rick Silverman, certainly has a lot of experience of taking these hit compounds, these lead compounds, as we call them, using tools of chemical synthesis to change them slightly, to develop a whole new generation of variants that then can go back through the high-put screening, produce something more effective, perhaps fighting greater than the initial symptoms, et cetera. So it's really a powerful project.
Exactly. So I think this is the way forward, because we need to find compounds that improve the health of diseased upper motor neurons and ask upper motor neurons whether they like it or not, right? Because the upper motor neuron degeneration is a hallmark of neurodegenerative diseases like hereditary spastic paraplegia, primary lateral sclerosis, ALS. It's not just one disease, all rare diseases, and improving their health is an effective treatment strategy because the movement starts in the brain, right? But so far, no compound has ever improved, their health, but there was really no preclinical test to investigate that, right? So this is why we collaborated with Dr. Silverman and to see if any of the compounds, improve. And two different mechanisms. One of them is the misfolded SOD1, and the other one is TDP pathology. So those are two distinct and non-overlapping pathologies. And it's very important that a compound or a group of compounds can actually improve them both at the same time. And our readout are on cellular assays that are translational, which are mitochondrial health, ER stability, apical dendrite stability, which we have shown that occurs both in the upper motor neurons in mice and upper motor neurons of patients. So that's why we think that for effective treatments, right, we need to lay the foundation for a cell-based and mechanism-focused drug discovery. So this publication was important for this because I think this was the first time that we were able to do this. And now, with our group of scientists, Chris, Baruch, and Omar, and with Sarah's help, we are trying to develop this high throughput or maybe semi-high throughput system where we can culture upper motor neurons that are diseased due to different mechanisms, right? And test different compounds. So far, we have maybe 17 compounds, but Dr. Silverman will be creating and generating, as you said, different variations of different variations, right? So we may end up having hundreds of them so that we begin to understand which compound is more effective for which mechanism, so that we can have numerous compounds and tackle the heterogeneity of the disease, right? So the expectation that one drug is going to cure everyone is over, right? And I don't think we should be waiting for Godot, right? Godot is not going to come. So we're not going to find just one miracle drug. There will be numerous drugs. And I think the high throughput system and the system that you described is the way forward, so that we can bring in very small amounts of cells into the system. And Baruch is actually very talented he's not using the machine, he's actually using his hand for the 96 well plates. And so far, we can plate them in those wells. And now with your help, hopefully we will determine the branching, arborization, axon outgrowth, all these measurements and in a, in an unbiased way and in a reproducible way. So I'm going to stop, sharing my screen. But I think these are exciting times for ALS drug discovery, because we identified the neurons, we identified the mechanism, we identified the translation, and we are lucky to have colleagues like you, and then core facilities and innovative leaders like you, so that, and also thanks to Along Swim and now thanks to DoD, we have support as well. I think this is the right time to make important discoveries, and I'm very happy to be with you today discussing those. Thank you.
Well, thank you, Hande, and I have to sort of echo some of the numbers that you're talking about in this molecular level analysis and things like that. It's overwhelming to see it as a layperson to consider. And I remember when NU9 was first introduced and announced and so forth, and really, I mean, potential game changer in ALS. And Rick Silverman told me that you had analyzed 50,000 different compounds before you had they sort of narrowed in on that. And it's sort of a mind blower to imagine in earlier generations of research the possibility of trying to analyze 50,000 anything. It's just so this is mind-boggling stuff, and the fact that it could be applied to neurodegenerative diseases and so forth, and that ALS could be a beneficiary of it is really, really overwhelming
Doug,
stuff.
50,000 is a lot, but pharmaceutical companies, the major pharma, they screen millions of compounds. But the key here, I think, my bias, is having the insights brought by fundamental research and this brilliant insight that Hande's group has developed to make a model system that is going to reflect this disease and all its various mechanistic variants. And it doesn't matter if you have 1 million compounds or 50,000 or 100. If you do not have a good model that reflects that disease, you're just wasting your time. And so her insights with the support of Along Swim is really leading the way and doing things that big pharma cannot do.
Right. Well, and it begins and ends with collaboration, and that is that's really the undercurrent of the whole thing, I think. So
we have some questions.
Yeah.
Okay, so Mukesh Gautam asks a question. He's an Along Swim fellow. It says, great technology. How many neurons can you plate in a well of 385 or higher well plate? Does the cell-cell contact affect the results, as there are a lot less neurons in such a well? Thank you. I don't know, how many cells do you put, Sarah?
That sounds like a Sarah question.
Hey, I thought this guy's from your lab.
Yes, it is from our lab. We are using not the 385, the 94 well plates, but in a much smaller. I think we would be using a much smaller volume, maybe, I don't know, 500 or 1,000 cells. Sarah, I don't know how many cells you use in those cells.
I don't know about motor neurons, but I've used Cortex before, and we do 5,000 in the 96. So probably, yeah, maybe 2,500
to 1,000, right?
Yeah, and the 384.
Yes. Okay. So Nicholas asks another question. One of paper mentioned, and one of your papers mentioned NU9. For the high throughput assay, is the library of compounds made of public compounds, Northwestern compounds, or other library that you pay for? I think, it was done by Dr. Silverman, so I don't know the exact answer to this, but I think it was, maybe you guys know.
Well, I think it was a custom library that was
library that he made,
created by him. Yeah, they were, again, intelligently designed specifically for this project, I believe.
Okay. Yeah,
that's great.
I think those were the questions.
Well, and we are, again, so grateful for everybody to tune in this evening. I've got pages and pages of notes here, and and so forth. I hope you got a lot out of it. Once again, I would invite everyone to a very fun swim this weekend. It's on Saturday. It's at Tower Road Beach in Winnetka, and all proceeds will end up at these fine researchers' labs. In fact, I see some familiar swimsuits from last year's swim. And not only can you register for this swim on our website alongswim.org, but marathon swimming doesn't usually attract a lot of spectators, but those are welcome too. And Tower Road Beach is a beautiful place to spend a Saturday morning. The other thing that is shamelessly obvious on the A-Long Swim website is a place to donate, and we will humbly accept any donations there as well. So thank you all. We're just at an hour, and I feel a lot smarter than I did an hour ago. Let me see if this chat is anything that that's from Kate. But thanks so much for joining us, and we'll see you the next time, and we'll see you on the beach in the meantime. Thanks so much.
And Curt, we will see you swimming this time as well. So Curt is one of the swimmers for A-Long Swim, and it would be nice to see you at the beach as well.
That's great.
Keep swimming, everybody.
There you go. Thanks, Curt. Thanks so much, Sarah. Wonderful work you're doing.
Thank you, Sarah. Thank you.